Let me welcome everyone to today's symposium, which is entitled Targeted GABA Receptor Modulation of Network Dysregulation in Depression. So what is depression, right? I mean, that's one of the questions that we're continually answering. And for many reasons, it makes sense to think about depression at a systems level. That is, instead of looking for a specific target located in the brain somewhere that's responsible for depression, it's really the discoordinated interaction of distributed brain regions that's responsible for depression, right? It's the individual getting stuck in a particular state, which reflects this kind of discoordination of dynamic functional connectivity. So as a metaphor, if you will, if one imagines a symphony orchestra in which the percussion is somehow 3 or 4 measures ahead of the rest of the orchestra, you're not going to get the kind of music that anyone's going to want to listen to. And similarly, those kinds of disconnections can lead to an individual being locked into a particular affective state instead of being able to transition between different affective states. As such, then, when one is attempting to understand what the source of that disconnection might be, an obvious candidate is GABA, because GABA is, as everyone knows, the major inhibitory neurotransmitter in the brain. It's responsible for setting the tone and for, if you will, for coordinating these interactions through tonic inhibition, acting in large scale to regulate the neuronal excitation throughout the brain. And there are lots of data suggesting that there are GABA deficits in depression. And more to the point, in this symposium, we now have ways of being able to target specific GABA receptors. GABA receptors are heteropentameric glycoproteins. They're composed of 5 different subunits around a chloride ion channel. And one can mix and match among 19 different subunits to produce very different pharmacologic and very different electrical properties. So today, what we are going to be talking about are these different ways of targeting the GABA system, and at a broader level, how will targeting GABA and in fact altering network interactions allow us to better bridge the gap between synapse and system in understanding depression. Our first speaker today then will be Etienne Sibille from the University of Toronto. And he will be talking about a specific type of GABA receptor, the alpha-5 receptor, and demonstrating its role in cognition and in neuroplasticity. Etienne?
Thank you, David, for putting this seminar together, this symposium, and for introducing us. I think, as you mentioned, there's a lot of renewed interest in the role of GABA in depression these days, and I think one of the reasons that this has come about also is, as you mentioned, we can be more specific in our targeting now, and we can also be more specific in how we can understand the whole system, you know, in terms of modeling and effect at the organism level. So I'm going to talk to you about novel molecule that we developed that targets specifically the alpha-5 GABA-A receptor. And so I'm going to give you a little bit of background, where we came from, show you the molecule, show you the data, and what the hope is to do with those molecules. So in terms of disclosure, you know, I have patent on the compound I'm going to describe. I'm also founder of a biotech company that we put together to actually bring those compounds to the clinic to go for cognitive deficits in depression. And below is my support. So over the past 10, 15 years, you know, work in my research group has led us to consider changes in the cortical microcircuit in the context of depression and other disorders. So on the screen here, you have, if you want, a canonical cortical microcircuit. You can think of it as a unit of information processing that is replicated across all cortical layers. And in this unit, it's formed by different cell types. Typically, you have this large excitatory glutamatergic pyramidal cells that propagate excitatory information, and that is under the control of different subtypes of GABAergic inhibitory neuron. And those inhibitory neurons can be characterized, if you want, by different features, such as what are the peptide they co-express, so somatostatin, VIP, or parvitamine, for example, or by electrophysiological property, and importantly also by which cellular compartment they target. So, for example, PV neuron are known to target the perisomatic region, therefore they regulate information output, if you want. SST neuron provide dendritic inhibition, therefore they regulate input, when we could say they participate in coding of information that is incoming, where VIP regulates mostly interneuron, they're also known to target pyramidal cell directly. So over the past, as I mentioned, 10, 15 years, we've come to realize that there seems to be a specific vulnerability in the context of depression of this particular subtype of GABA neuron, the SST. And I'm just showing you data. So we published, multiple findings on this. This has been replicated by other groups. But here in 3 frontal cortex regions, DLPFC, ACC, and amygdala, for example, 3 different studies where we show that there's a decrease in expression of this peptide, in different nuclei of the amygdala, across layer in DLPFC, and across layer. And whereas changes in PV and VIP are minor or non-existent in the same studies. So this suggests, SST, of course, is a neuropeptide that is expressed in the cell, but the primary function of those neurons is inhibitory. They co-express GABA and mediate their function through GABA-A receptors. This decreased somatostatin is actually not, not a novel finding. We published a couple of years ago a little survey, across disease that all the way back to the 1980s in the context of Alzheimer, for example, reduction of expression or density of SST expressing cells has been reported. And you can see there's a really great consistency in terms of direction of effect. We're providing a lot of information on depression, but schizophrenia, bipolar, including neurological disorders such as Parkinson. I'm not going to talk too much about that, because this is a short symposium, but just to give you an idea, the reason why we believe this is so widespread is that there's an intrinsic vulnerability of those cells to biological adversity. We've shown that it's BDNF dependent and it's related to some of intracellular mechanisms that are unique to somatostatin neurons. So this explains maybe the fact that we see them everywhere. It's because if you want, those cells are somehow weak link in the brain. This is also, as David mentioned in his introduction, in the context of many levels of information across scales of reduced GABA function in depression. Going back, to low GABA level in CSF, low tissue level measured by magnetic resonance spectroscopy, the Jerry Sanacora et al. Some of the early human postmortem studies reported decreased markers of GABAergic neuron that overlap with those SST. You can measure, you can do neurophysiological measure using TMS, reduce, etc. And these have been linked to some extent to some symptoms. I'm linking here anhedonia in depression. In the context of Alzheimer, reduced SST neurons had been initially correlated with cognitive functions. So there's a broad context. What I think our findings provide is a little more specificity in this broad GABAergic context is that if we go back to our cortical microcircuit, what I have here outlined in those dashed lines is that we believe that this part specifically, the somatostatin neuron that provide dendritic inhibition in cortical layers, mediated by the alpha-5 GABA-A receptor mostly, and I'll come back to that in a second. So we think this particular pathway is affected, so in terms of both reduced expression, neuron number in some cases, postsynaptic receptor. I'll come back to the end a little bit, but this is supposed to actually affect excitatory signal to noise ratio onto a pyramidal neuron in terms affecting the coding and processing of information, suggesting a potential role in cognitive dysfunction. So that was a hypothesis that was supported by several line of results from multiple groups, but we followed more specifically on that particular line of research. In terms of, so that was, if you want, human postmortem study evidence, primary evidence in human sample. In terms of causal role, we can turn to, of course, genetic and other environmental manipulation in mouse models. And here the data, I'm just lining up some results from our group, from others as well, showing that, you know, if you either use optogenetics or DREADS approach to reduce the function of somatostatin cells, or if you use knockout of genes, in general, you know, if you affect this particular pathway that I showed you before, you know, you can observe in mice, you know, sets of symptoms at the behavior, neuroendocrine, neurophysiological, and symptom level that would be, if you're analogous of what you see in a depression syndrome in human. And importantly, also, it's been shown that, you know, if you, you know, for example, there's a group from Josh Gordon's group, it's at NIMH, you know, it showed that if you use opto to reduce the function of cells, you do affect spatial working memory. The Furman lab in Germany has shown also that in the context of Alzheimer's disease that I mentioned before, the reduced function of those SST cells is specifically associated with memory deficit. So the summary of this is that, you know, there's a causal role there that is suggested by those studies in addition to the correlative findings that we see in human. So based on that, you know, the fact that this pathway up there that I'm showing on the left, you can see my cursor, by the way?
Yes.
Okay, good. So what I'm pointing at here, this pathway, you know, the function of somatostatin cells is in large part mediated by the alpha-5 GABA-A receptor, which lead us to the structure of this receptor that you'll hear a lot throughout this symposium, and we'll do it very quickly. You'll hear more again later. Typically, it's a pentamer, it's a chloride channel that is formed, in this case, by 2 alpha, 2 beta, and 1 delta subunits with different binding sites for either GABA or for modulators. The most famous modulator, of course, are benzodiazepine that bind at the intersection of the gamma-2 and the alpha subunits of the subtypes, the 1, 2, 3, and 5, more specifically. The problem is that those compounds, due to the pan-GABA-A receptor activity, have, you know, they're very robust anxiolytic effect, but they also have multiple side effects, which is thought to be mediated by the fact that, you know, the alpha-1 mediated effect is supposed to mediate some of the sedation effect, etc. So it's always been thought that if we can be a little more specific, if we can get to really the next generation of benzo that are much more specific to a subtype, we may actually discover novel therapeutic purpose for augmenting GABA-A receptor function. So based on what I have described, we've focused on this GABA-A, alpha-5 GABA-A, and I think what is interesting also compared to other subunits, they have a very limited, very restricted anatomical restriction. I told you they're located on dendrites of pyramidal cells, but they're also restricted mostly to hippocampus, cortex, and some minor expression in other brain regions. So that's of interest if you're in drug development because you don't have widespread expression throughout the body, therefore limited potential for side effects. So in our case, we worked with Jim Cook, a medicinal chemist at the University of Wisconsin. We to generate novel compound that would preferentially activate alpha-5 GABA A receptor. We published that. So, you know, basically we started with a safe chemical backbone combined of flumazenil and diazepam, because it already binds those pockets, and did some chemistry to modify some of the residue to, if you want, achieve the goal of preferentially activating alpha-5. On the right here, you have some cell EPHIS activity, where you saw, for example, this is activity alpha-5 versus the other subunit, alpha-4, alpha-6 have no activity, which we expect. And we have different compounds with different profiles that we've been tested. And we know those compounds pass the blood-brain barrier. They're safe. We've done a lot of, if you want, PK, pharmacokinetics and toxicity study on those stuff. At the behavioral level, I'm showing you, as expected for derivative of benzo, you know, and here you have the diazepam as a comparison. In the elevated plasmase, you can see systematically dose response, anxiolytic effect, increased exploration of the open arm. This GL2, 7-3, 7-4, they're just different compounds that we've tested. And again, diazepam show the same effect. You start to see a dissociation with just, you know, this potentially, you know, controversial test, if you want. It's just a screening test for response to an acute stress. The 4-stream test where diazepam doesn't have an effect, but you can see you have this nice reduction in immobility with that compound. In this case, suggesting potential antidepressant activity. In terms of cognition, which is really what we're interested in. What I'm showing you here is an assay, the Y maze. You know, this is a test in which a mouse learns to alternate between one arm and you know, typically the mouse will enter this maze, explore one arm, you will take it out, leave it out for 30 to 90 seconds. When you bring it back, it will explore the other arm. This spontaneous alternation, and we do that 6 or 7 times in a row, and you know, there's a kind of a cognitive load that increase, and typically a mouse will very rapidly behave at, you know, 80 to 90% accuracy in this test. What we show, if you do a simple daily restraint stress, you know, the classical chronic restraint stress, after 1 week, you can see that a mouse will perform at, you know, 50 to 60%, which is not too much different from random chance. So just chronic stress is sufficient to affect spatial working memory. And here I'm showing you with the specific compound called GL273, our lead compound, that we can dose dependently reverse those cognitive deficits acutely. This works in male, in female. We've replicated that multiple times. Using a more relevant model, if you want, it's called unpredictable chronic mild stress, where mice are exposed to variable stressors over a period of 4 to 6 weeks. We can see that similarly, after 4 weeks of chronic stress, mouse will show a deficit in spatial working memory, which interestingly we can reverse acutely or chronically. So this, I'm sure you're all aware of this, you know, acute or fast effect that we look in the context of depression. This is not a fast antidepressant, but it's a fast pro-cognitive effect. So we can acutely reverse those cognitive deficits after a single injection or after daily injection for a week. So next, we were actually interested, apologize for the poor definition of this image, but we're interested to know so what it does at the neuron level. And here we use the Golgi approach. And what we show you is that after chronic exposure to GL, we know that, first of all, in chronically stressed animal, it is known that dendritic length, spine density, spine number goes down. This is shown here. So this we replicated multiple findings from the literature. What we show here is after 4 weeks of treatment, we can actually reverse, or in this case would be block, if you want, the development of this cellular phenotype. So we reverse changes in spine spine dendrite length and spine number, and we do that both in the frontal cortex as well as in the hippocampal CA1 region. So it's interesting because we have symptomatic effect, but we really also potentially target the underlying pathology, or contributing pathology at least. Next, I want to switch to a different paradigm, which is this pathway is also known to be affected in the context of aging. I mentioned that earlier. Here, I'm just showing you some data, the expression of somatostatin decrease. You know, these are two regions of the frontal cortex, very robust downregulation of expression of this gene. You can see here across subject, you have this really robust downregulation from the age of 20 to 60, 70, 80 in human subject. Same, the postsynaptic receptor, GABA5, the gene coding for this subunit. Similar downregulation, and by the way, those two correlate to a level, I think, 0.7 or 8. So there's really strong correlation, so suggesting that they really co-occur within subject. We've shown also in some other study that in some case, you know, in a very old subject, you may have actually a reduced density of somatostatin neuron. The point is the same pathway that I showed you affecting depression is robustly affected in the context of normal aging, in addition to Alzheimer and others. And there have been some reports that, you know, again, alpha-5 PAM, those positive allostatic modulator at the alpha-5 receptor I showed you, may have some pro-cognitive effect in aging. So we tested that, and the same assay that I showed you before, we can acutely reverse cognitive spatial working memory deficit in old mice. We're talking about 22 to 24 old mice. So we can acutely reverse those deficits or after chronic treatment. And I just mentioned here the chronic treatment is interesting because, as you know, GABA-A pharmacology is sometimes limited by desensitization or habituation to the effect of chronic targeting. But here we show that, and may have to do with the kinetics of the alpha-5, but that the effect is sustained after, in this case, 10 weeks, 8 weeks. We've done up to 3 months. Going at the Golgi level, similar as I showed you in the context of stress model, we can see that after, in this case, 2 months of treatment, we can actually reverse the reduced length of dendrite and spine number that are typically seen in an old mice. So you see it here. You have, you know, reduced length and reduced spine number, and we can actually, if you want, rejuvenate those neurons to some extent. And I don't have that slide by the way here, but those correlate with the cognitive function. Those measure, you know, because this was done in the same animals. And in this case, it's also interesting because it's only shown in the distal dendrite, which is really where the alpha-5 receptor is located, as opposed to basal dendrite. And again, we've shown that both prefrontal and hippocampus. Moving on, we just a couple more slides. We were also interested to say, well, since there was a neurotrophic effect, and you know, there's a lot of interest in reduced neurogenesis in the context of those illnesses, we show here just the first study done with the lab of Denis David at the University of Paris Sud, that just in normal mice, 129 SV, because they have a low neurogenesis, we can see that we can actually increase neurogenesis after 4 weeks of treatment. To a similar extent, increased cellular proliferation and increased maturation index of neuron, suggesting, you know, and fluoxetine here is used as a control, maybe not to the same extent, but still, it's quite interesting that, and still not really well characterized at all, that increasing inhibition can have neurotrophic effect, as I showed you in the previous slide, but also neurogenic effect. Typically, increasing GABA has either no effect or actually could be detrimental. So again, here we think because it's more specific, targeting alpha-5 on dendrite, that we have this effect. So in summary, I show you that the pathway that's therefore, that mediate dendritic inhibition appears to be vulnerable to biological adversity in the context of depression and other illnesses. Brain disorder, aging converge on impacting this pathway. And also showing that augmenting this pathway, in this case with molecules that target the alpha-5, we can improve cognitive and mood symptoms in depression and aging model, but also we can target the underlying or contributing cellular pathology. Since we're not, I just have a couple more slides. Since we're not bound by time, I'm going to go through them, try to maybe tell you why, what do we think is happening, you know? So yes, we have those really nice kind of symptomatic effects, but what is the model? How does it work? So I mentioned earlier that, you know, those neurons, you know, SST neurons are thought to regulate signal to noise ratio. And that is, in a healthy circuit at baseline, those cells have relatively high firing rates. And they mediate, if you want, they shut down pyramidal neuron in layer 2, 3. So what we see is the pyramidal neuron is typically the sparse activity. When stimulus comes in from any sensory modality, SST neurons are typically shut down by VIP neurons, which allow information, excitatory information to get to neurons. So you have this high, suddenly excitatory activity in contrast to a low activity on the baseline. So the signal to noise ratio is thought to contribute to information coding processing. In a pathological microcircuit, we have, if you want, weak inhibition by somatostatin neuron, which result in increased baseline activity. When information comes in, we probably have a very similar pattern than in a normal, healthy microcircuit. However, the signal to noise ratio now is much lower, therefore reduced capacity for coding and sensing relevant information. This is summarized to the right. If you look a little broader, you know, the somatostatin neuron have been characterized as providing, if you want, a blanket of inhibition. You know, they really shut down the system and the activity. And as I mentioned, when signal comes in, it leaves out very specific information, and those green arrows would be information that we expect. Here we see that this blanket is loosened in the context of depression. Therefore, when signal comes in, you know, so you have a temporal but also spatial signal to noise ratio where this information becomes more difficult to detect compared to this high background noise, both spatially but also temporally. And I'm just putting this here, you know, with the PAM effect, positive allosteric, we're thinking that we're tightening the inhibitory blanket, therefore restoring signal to noise ratio. There's been a lot of data on alpha-5 negative allosteric modulator showing antidepressant effect, fast antidepressant effect. The idea here is that it actually loosens further the inhibitory blanket. So maybe under condition where high volume of information may be okay, such as, you know, fast flight response, etc. So some of these assays, this may have antidepressant effect, but we believe that this is a more targeted approach to follow. So based on that, I just want to thank the whole team. This has been a collaborative effort over the past few years. Specifically to thank Thomas Prévost in my group, who's been leading a large part of this effort, and our collaborators, University of Wisconsin. So thank you very much. Appreciate your attention.
That's great, Etienne. Thank you very much. I'm sure there will be a number of questions. It's really a lovely story you've put together.
Thank you. We're
going to capitalize on a couple of themes that you raised with our next speaker, Jamie Maguire from Tufts. You raised the issue of stress and its effects on neural signaling. Jamie is going to talk some about stress, but will focus more on a specific subtype of GABA receptors. That is those that contain the delta subunit, which plays a critical role in tonic inhibition, and which is preferentially bound by neurosteroids, which are also stimulated by stress, unlike benzodiazepines, which do not bind delta-containing GABA receptors. Jamie's created some wonderful behavioral models that involve both stress and inactivation of delta subunit containing GABA receptor signaling. And she's going to tell us a little bit about that now in her work with neurosteroids. Jamie?
Thank you, David. I just want to thank you for organizing this panel and thank the rest of the panel for being willing to come together virtually to present on this topic. And so, as David mentioned, I'm Jamie Maguire, and I'm an associate professor in the neuroscience department at Tufts University School of Medicine. And I'm going to share with you some of our data demonstrating a bidirectional effect of stress and neuroactive steroids on network and behavioral states, and what we think is relevant to this concept of effective switching. So first, I have a conflict of interest statement. I serve on the scientific advisory board for Sage Therapeutics, and I have a sponsored research agreement also with Sage Therapeutics. So what I'm going to go through today is really the inspiration for these studies, which were in clinical trials with brexanolone and some of the later compounds developed by Sage Therapeutics, which show persistent antidepressant effects. And these effects aren't very well explained by the known mechanism of action of these compounds on GABA-A receptors. And so we began to think how what could be underlying these more persistent antidepressant effects, and started thinking, as David nicely introduced in the introduction, this idea that perhaps these compounds are inducing a state change, and that state change is more stable and persistent and could contribute to these prolonged antidepressant effects. And so in order to start investigating that hypothesis, we started trying to understand more deeply how network states are shaped within outside of the hippocampus, so focusing on the basolateral amygdala and emotional processing. And we also were thinking about how risk factors, such as chronic stress, could negatively impact these circuits, cause disruption in these behavioral and network states, and then looking at the possibility that allopregnanolone may be able to restore this healthy network state, and building on this idea that allopregnanolone might be a mediator of effective switching, which was actually introduced by David Rubinow. So I just wanted to show you some of the data that got us thinking along these lines from the clinical trial with brexanolone, now trademark Zulresso, which is an allopregnanolone analog. And what was shown in the clinical trials is that there's this really rapid antidepressant effect that is very exciting and unique, but also that there are these persistent antidepressant effects. So at the end of treatment at 60 hours, they're seeing sustained antidepressant effects out to 30 days in this study, which is now out to 45 days in some additional studies. So again, we began thinking about this idea that perhaps there's a change in the network state that is underlying these changes in behavioral states, and thinking about what that means as far as the episodic nature of depression. So we don't really understand how these episodes of depression emerge and manifest, and is there a way that we can study that at the network level? And so again, we were really inspired by David's idea that allopregnanolone could be a mediator of effective switching. And we wanted to build on this concept to see, can we understand this at the network level? And there was some precedent for looking at network level activity as far as behavioral outcomes. And a lot of this data is coming out of the fear learning field, where specific changes in the pattern of network activity, so oscillations within specific brain regions, correlate with different behavioral states and fear and safety states. And it's not just the activity within that region, but it's really the way that oscillations are used to communicate between regions and in between the prefrontal cortex and the basolateral amygdala is important for signaling either fear or safety. And it's thought that these oscillatory states are really the substrate for fear learning. And I got involved with this through a collaborator at Tufts University, Leon Reimers, who is interested in fear learning. And we began to do in vivo recording, looking at oscillations associated with different behavioral states. But outside of fear learning, we really were interested in whether this concept is broader than just fear learning and whether it could be more applicable to emotional processing more broadly. So in order to explore this, there's some unanswered questions that we wanted to tackle. One of them is people were doing these in vivo recordings in the basolateral amygdala, and it wasn't even clear whether the amygdala could generate oscillations or whether these oscillations were entrained from the hippocampus or other regions that are known oscillators. And then if we do see that the basolateral amygdala is capable of generating oscillations, what mediates the transition between oscillatory states? And this is really important for understanding what could be potentially relevant to the episodic nature of depression. In order to understand the underlying neurobiology, we need to understand how the network and how the behavior transition between states. And our major question is whether neuroactive steroids can facilitate modulation between behavioral states and with the hypothesis that it may work to transition to the healthy network state and that state may be more stable. And finally, if chronic stress, a major risk factor for depression, can disrupt these network states, and again, if so, can neuroactive steroids combat that and restore the healthy network state? So to answer the first question, we wanted to just determine if and how oscillations are generated in the basolateral amygdala. And we focused on an ex vivo prep where we could isolate the basolateral amygdala. We actually physically removed the hippocampus and looked to see whether we could generate oscillations in the basolateral amygdala. And what we find is that the basolateral amygdala is capable of generating oscillations. Here I'm showing you gamma oscillations that occur, with a peak frequency around 40 hertz. We then look to see what is the mechanism of this oscillation generation to understand more how these oscillations are generated. So the first thing we did was look to see whether excitatory neurotransmission is required for generating these oscillations. So we applied the AMPA receptor antagonist GICI, and what we found was that the blocking excitatory transmission was unable to alter these oscillations. However, if we inhibited GABAergic inhibition with gabazine, what we found was a very rapid collapse in these oscillations. So what we've determined is that unlike the hippocampus, the basolateral amygdala is an ING network. So it's an interneuron network gamma oscillator in which these interneurons drive oscillations in the basolateral amygdala. And so we wanted to look at what types of interneurons are driving these oscillations. So we injected channel rhodopsin into the basolateral amygdala of parvalbumin Cree animals and looked to see whether parvalbumin interneurons are capable of generating oscillations in the BLA. we can stimulate at different frequencies, 5 hertz frequency or 30 hertz frequency, and we can entrain oscillations in the basolateral amygdala just by activating parvalbumin interneurons. And we can do that across frequencies. So now we've shown that there's a critical role, and I'm not showing you here, but if we silence parvalbumin interneurons, you can completely collapse oscillations in the basolateral amygdala, showing that parvalbumin interneurons are really critical for oscillation generation in the basolateral amygdala. So now that we know more about the mechanisms driving these oscillations, we want to know how they transition between oscillations. And given the critical role of parvalbumin interneurons, we proposed that inputs that could modulate parvalbumin interneurons can alter oscillations in the basolateral amygdala. We think that those inputs are probably pretty diverse, and it's a major focus of my lab right now is to understand the inputs onto PV interneurons in the BLA, to understand how different inputs might be able to modulate these oscillations under different behavioral states, but also under different pathological conditions. And I think that this really speaks to the heterogeneity that we think about in the patient population with depression, is that there's probably many, many ways to disrupt network states. And what we're trying to understand is the nature of that disruption and how we can restore healthy network function. So one of the things that my lab focused on and was interested in is that these parvalbumin interneurons in the BLA express a high density of these delta subunit containing receptors. So here you see this dark delta staining in the basolateral amygdala, which is lost in animals that lack the delta subunit just on parvalbumin interneurons. So they're really co-localized with parvalbumin interneurons in the basolateral amygdala. I won't go into too much, but building on what Etienne was presenting, is we're really interested in the subcellular distribution of these receptors, which is different in interneurons than it is in principal neurons. In interneurons, the delta subunit is largely expressed somatically, and in principal neurons, it's expressed primarily in the dendrites. And we think in these two different cell types that the delta subunit is subserving a different function. And we were interested in whether in the interneuron population in the basolateral amygdala, it may be involved in coordinating oscillatory states. So just to give you a little bit of an introduction into our interest in the difference between these delta subunit containing receptors and typical phasic versus tonic inhibition, our lab largely focuses on the role of gamma-2 containing GABA-A receptors, which mediate phasic inhibition, are localized in the synapse, confer benzodiazepine sensitivity, and are unique from those that contain the delta subunit. So receptors that contain the delta subunit largely mediate tonic inhibition. They're either peri or extrasynaptically localized, and important to the talk today, they confer neurosteroid sensitivity. So the fact that these parvalbumin interneurons express at a high density these delta subunit containing receptors, we thought that there might be a role for tonic inhibition in controlling the pattern of activity within the BLA. So just so that we're all on the same page, I want to introduce the difference between phasic and tonic inhibition. In response to a single vesicle being released from the presynaptic terminal, you get a small number of postsynaptic receptors that are activated and a small postsynaptic current or miniature postsynaptic current. But in response to an action potential, when many more vesicles are released, there's a larger number of postsynaptic receptors activated and a larger postsynaptic current. And that is what we consider the phasic component of GABAergic inhibition. Whereas tonic inhibition, these peri or extrasynaptic receptors can actually respond to ambient concentrations of GABA. And the only way that we can resolve the contribution of these currents is by applying a GABAergic antagonist. And what you can see is with the addition of the GABAergic antagonist, you get a shift in the holding current. And that shift in the holding current is attributed to this tonic or persistent form of inhibition that was present in the background. And so we're still trying to understand what exactly these two different types of GABAergic inhibition are doing functionally. But we can use this to look at the contribution of delta subunit containing receptors to the regulation of principal neurons versus parvalbumin interneurons in the basolateral amygdala. And so using electrophysiological recordings, these cells are held at minus 60 millivolts, and we apply allopregnanolone and look at the allopregnanolone sensitivity of the tonic current, which can be revealed by the addition of gabazine or SR95531. And what we find is that there is a small tonic current in principal neurons. But if you look at the potentiation by neurosteroids such as allopregnanolone, we see sensitivity almost exclusively in parvalbumin interneurons in the basolateral amygdala. So we think that this sensitivity is conferred by these high expression of the delta subunit containing receptors on parvalbumin interneurons that we don't see on principal neurons in the BLA. So we propose that the tonic inhibition on parvalbumin interneurons may help control the transition between synchrony in the basolateral amygdala. So we know that tonic inhibition has been proposed to act as a high-pass filter and really modulate the gain and enhance fidelity of information processing. And so when we think about in the presence of a low tonic inhibition, we're going to get asynchronous activity transmitted throughout the network. So these parvalbumin interneurons can connect to hundreds, if not thousands, of principal neurons in the basolateral amygdala. And so it has the capacity to control the output of a large number of neurons. And in that way, we think can really control synchrony of the network. Now, in the presence of neurosteroids, which can increase tonic inhibition, we think that in this case, you're only letting through the superthreshold events, and now you're more able to synchronize the network at this specific frequency. And this has actually been beautifully modeled by Ivan Pavlov, who is in Alexei Semenyov's lab, and demonstrated that tonic inhibition can bidirectionally control interneuron firing frequency and therefore synchrony within the network. And this was looking at tonic inhibition of interneurons in the hippocampal network, but we think the same principle holds for the basolateral amygdala. And what they show is if you have no tonic inhibition, you basically have no synchrony in the network. If you have a low or moderate amount of tonic inhibition, you are able to synchronize the network more effectively. But if you push the inhibition too far, you begin again to destabilize the network. So there's this role of tonic inhibition in optimizing network synchrony. And so we think that this tonic inhibition on parvalbumin interneurons in the BLA may be playing this role in subserving this function to coordinate network activity, and in that way can control the transition between different network states. So since we know that neurosteroids can be synthesized in the brain, either they can come from the periphery or be synthesized locally in the central nervous system and act postsynaptically to enhance tonic inhibition on the postsynaptic neurons, we propose that neurosteroids may be capable of transitioning between network states. And that's exactly what we aimed to test. So in order to do this, we implanted electrodes for local field potential recording in the basolateral amygdala. And we injected animals with allopregnanolone. And what we found is that if you inject animals with saline and compare the network response to allopregnanolone, we see that allopregnanolone is capable of altering the network states within the basolateral amygdala. And that in part involves delta subunit containing receptors, because we see that this effect is dampened in mice that lack the delta subunit containing receptors. We can look at this effect across frequencies, and what we see is an effect of allopregnanolone on, particularly on theta oscillations in the basolateral amygdala, which is again mediated in part by delta subunit containing receptors. So interestingly, we saw this across species. So working with Sage Therapeutics, we were able to look at the effects of Sage 516, which is one of their tool compounds, which is very similar to allopregnanolone. And we see that there is the ability of Sage 516 to potentiate these oscillations in the theta range in the basolateral amygdala. We see a similar effect in both rats as well as in patients that were treated with brexanolone. We also see that the effects of allopregnanolone are unique to that of other GABA agonists, such as benzodiazepines. If we look at the effects of diazepam in both mice and rats, what we see is that we do not see this potentiation in the theta range that we do with both allopregnanolone and Sage 516. So these effects in the theta range are unique to neuroactive steroids and unique from other GABA agonists such as benzodiazepines. So we wanted to see whether allopregnanolone acting in the basolateral amygdala could also influence behavioral states. So we implanted mice with a cannula placed in the basolateral amygdala, and we infused allopregnanolone 30 minutes or vehicle 30 minutes prior to behavioral testing. And what we found is that this administration of allopregnanolone does not have a sedative effect. When we look at the locomotor activity in the open field, we don't see any effect of the allo infusion on locomotor or exploratory behaviors. But if we look at tests for anxiety-like behavior, such as the light dark box, what we find is that mice, wild-type animals that are infused with allopregnanolone, exhibit an increased amount of time that they spend in the light compartment of the light dark box. And again, this is in part due to delta subunit containing receptors, because we don't find this in delta receptor knockout animals. We see a similar effect in the elevated plus maze, where infusion of allo in wild type animals increases the amount of time spent in the open arm, an effect which we do not see in delta receptor knockout animals, suggesting that BLA exerts an effect, allopregnanolone exerts an effect on anxiety-like behaviors, specifically by acting just in the amygdala. We also looked at depression-like phenotypes using a tail suspension test, and found that mice that were infused with allopregnanolone had a decrease in the amount of time spent immobile in the tail suspension test. And again, we did not observe this in delta receptor knockout animals. So we see that allopregnanolone can affect behavioral states through actions in the basolateral amygdala. But what we wanted to see was, we're looking now at acute effects on the network and acute effects on behavioral states. But what we want to know is this relevant to the prolonged antidepressant effects that were observed in clinical trials with neuroactive steroids? And are neuroactive steroids capable of inducing these persistent change in network and behavioral states in our mouse models? So similar to the paradigm that Etienne introduced, is we used a chronic unpredictable stress paradigm for 3 consecutive weeks. Typically, the behavioral testing occurs pretty shortly after the last bout of stressors. And we find if we test the animals 24 hours after the chronic unpredictable stress paradigm, this is just one behavioral test showing a depression-like phenotype, where we see a decreased latency to immobility and an increased total time immobile in the tail suspension test. And we see that same thing in the forced swim test. But what about the persistent effects on behavior? So we did the following experiment where we recorded local field potentials in animals, naive animals, 24 hours before subjection to chronic unpredictable stress, with or without treatment with Sage 516 or allopregnanolone. We then recorded changes in the network activity at 24 hours after the chronic unpredictable stress and once a week for 4 consecutive weeks. And what we found is if we looked at behavior after that 4-week period, we still see an effect on the behavioral state of these animals that were subjected to chronic unpredictable stress, where there's a decreased latency to immobility and an increased total time immobile in the tail suspension test compared to wild type, which is indicated, home cage controls, which are indicated by the dotted line here. If you treat the animals with Sage 516 during the chronic unpredictable stress paradigm, you can blunt the behavioral effects of chronic unpredictable stress. So we see a protective effect on behavior with Sage 516 or allopregnanolone treatment. Well, what about the network states? So we looked at the local field potential, again, before and 4 weeks after the subjection to chronic unpredictable stress with allopregnanolone or Sage 516 treatment. And what we find is that even after 4 weeks post chronic unpredictable stress, the animals that were treated with allopregnanolone show a reduction in the change in their local field potential compared to baseline pre-CUS exposure. If we look at the correlation between the changes in network activity and the total time immobile, what we find is that there's a relationship between the change in the local field potential in the basolateral amygdala with the change in the behavioral state, where the larger change compared to the pre-CUS is associated with increased depression-like phenotype in the tail suspension test. And you can see here in the orange is the Sage 516 animals in this cohort, which have a smaller change in the local field potential, and they're less affected behaviorally, which you can see in the average data here. So what I've shown you is that there's a role for parvalbumin interneurons in modulating or generating oscillations really in the basolateral amygdala, and that these PV interneurons express a high density of the delta subunit containing receptors, which we think is a site of action to modulate parvalbumin interneurons, modulate network states and behavioral states. And we've shown that allopregnanolone is capable of altering oscillations in the basolateral amygdala, as well as behavior through actions in the basolateral amygdala, which are at least in part mediated by delta subunit containing receptors. We show that the effects of allopregnanolone on the network are unique to those of other GABA agonists such as benzodiazepines. And finally, our work is beginning to look at these persistent antidepressant effects on network and behavioral states, and showing that allopregnanolone or Sage 516 is capable of well, one, chronic unpredictable stress is capable of impairing the network, which is associated with behavioral abnormalities, and that allopregnanolone or Sage 516 are capable of preventing network dysfunction and decreasing deficits in the behavioral states. So getting back to this concept that David Rubinow came up with, that allopregnanolone may be involved in mediating effective switching, we're now seeing at the network level that allopregnanolone is capable of shifting between network and behavioral states. And finally, I'll just thank my lab. And again, I want to thank Panelise Antonio-Dayo, who is an incredible postdoctoral fellow, now research associate in my laboratory, who has developed unbiased data analysis tools for looking at these shifts between network and behavioral states. Okay.
Jamie, absolutely fabulous story, and really just a wonderful systematic delineation of observations that you've made, 14 years ago. So really fabulous. Again, using your work on oscillations and network communication as a way of getting from synapse to behavior. Our next speaker, John Murray from Yale, will depart a bit from the GABAergic theme, but will talk about computational methods of really being able to understand relationships between local synaptic activity and local field potentials and more systematic network interactions. John?
Okay, let me share. Yeah, I'm really excited to participate in this symposium, and it's been really fantastic. The prior two talks really describing kind of microcircuit cellular synaptic level dysfunction and how that may induce kind of dysfunction at the level of circuit level computations and ultimately psychiatrically relevant symptoms. So my lab is a computational neuroscience lab, and so what we focus on in terms of computational psychiatry is mathematical models of neural circuit dynamics at different levels of analysis, and then relating those through data analysis in collaboration with experimentalists to different levels of analysis in terms of experimental data. And so today I'm going to talk at a bit of a higher level, linking all the way to human neuroimaging beyond kind of the more detailed microcircuit level. So my disclosures, I consult for Blackthorn Therapeutics around biomarker development and have a sponsored research agreement from Blackthorn as well. So I'd like to start with the acknowledgements. This is primarily the work of a physics PhD student in my lab, Joshua Burt. And it was really primarily a collaboration between my lab and the lab of Alan Antichevich at Yale and Katrin Preller and Franz Vollenweider at University of Zurich. Okay, so the kind of big problem that we struggle with in computational modeling is how to bridge these levels of analysis. And I really see that as the purpose of developing computational models, to try to formalize what those links are. And in psychiatry, especially in terms of therapeutics, it's very vast, right? Where we are looking at symptom variation at the individual level, at the in terms of behavior and how that varies, and then ultimately what we want to address that with is therapeutics at the level of molecules and pharmacology, right? And so this is a very large gap to cross. And so we can start to kind of fill in the pieces and think about how we can use modeling to try to help that. And so, and really there are kind of two motivating questions for us in terms of making this gap. One is that if we have a molecule, in terms of psychiatric indications and outcomes, how do we know what to apply it to, right? And conversely, if we do not have a therapeutic, how can we identify for a given symptom dimension or psychiatric impairment what may be promising molecular level targets that we can go after? And so, one way that we can start to close this gap is by linking symptom variation to, say, neural biomarkers. And there's huge efforts in terms of large consortia, imaging patients with fMRI, EEG, MEG, et cetera, to try to establish what, at the level of large-scale neural circuits, are altered in relation to psychiatric disorders and dimensional impairments. And this is still a very large gap to then think about how a molecule could bridge all the way to the level of kind of whole brain systems level alterations. And so this is where we're trying to kind of bridge this gap with modeling. And so we can take one step further, going from a particular molecule and think about the receptors that it targets and the distributions of those receptors, which we could map, for example, through transcriptomics, looking at the differential patterns of gene expression at the level of cells and circuits and brain regions at the whole brain level even. And in particular, today I'm going to focus on differential expression patterns for receptor-related genes across brain regions. We could then use that information to link all the way to, say, neuroimaging measures, comparing at the level of large-scale brain regional topographies, And then what I'm going to talk about today in terms of modeling is thinking of a target not of the clinical neuroimaging data, but of using pharmacological neuroimaging as a more controlled and precise target where we have some idea causally of what receptors and cell types might be mediating the neuroimaging alteration. And then finally, and again, we can kind of try to relate these two levels of analysis. And then with computational modeling, the idea is that we're simulating at some level of resolution neural circuit dynamics, which can be informed by neuronal and regional distributions in receptors, for example, from transcriptomics, and then make predictions at the level of whole brain neuroimaging data, which we can test and validate with pharmacology and ultimately bridge to the level of clinical data and individual variation. And so this is the kind of big picture view of how we're trying to cross all these levels of analysis. And I'll today kind of tell you a story about how we're trying to do that. Okay. And so again, the kind of main thing from this is focusing on the level of circuits and systems in order to bridge these levels of analysis from molecules to behavior. Okay, so one source of data that we're using is gene expression mapping in the human brain. And for this, we're leveraging the Allen Human Brain Atlas. And so this is RNA microarray measurements from 6 postmortem brains. And we run that through our neuroimaging pipelines, actually, to arrive at group level, parcellated, surface-based maps in the same space as our neuroimaging measures. And so this allows us to kind of quantitatively relate these transcriptomic measures in terms of brain-wide regional expression differences to the brain-wide patterns of neuroimaging data. So they're kind of operating in the same space, which ends up being very important. And to give kind of an example of what we get out of that relevant to this symposium, we can look at, for example, GABA-related genes. And so, for example, here I'm showing for 2 genes which code for the alpha receptors, GABA-A receptors, the alpha-1 and alpha-5 subunits here shown on the left and right. And what you can see is a very different topographies. So the alpha-1 gene is primarily expressed in sensory regions. You can see in visual, auditory, somatosensory, whereas the alpha-5 has low expression values in sensory, and it's really more strongly expressed in association regions such as insula, temporal, and prefrontal cortex. And we can also look at, for example, these are calcium-binding proteins, as shown in the previous 2 talks, which are interneuron cell type specific markers. And again, we see very different patterns in the expression topography. So the p-valve, which is a marker of parvalbumin interneurons, again, high in sensory, low in association, and SST shows an opposite pattern. And kind of there's a correspondence between the alpha-1 and p-valve and the alpha-5 and SST, as Etienne described in his talk. So the idea is that we can really kind of use these large-scale gradients to now think about targeting brain regions and networks and systems through pharmacology, because the effect of pharmacology is not uniform across the brain. And therefore, if we see preferential alterations in different brain regions, we can think about getting regional specificity through a systemic pharmacological manipulation. So the question is, how can we kind of use this and build up mechanistic models? And so previously we had analyzed this gene expression data and was really looking at the question of gene expression specialization across the cortex. And so there's this idea of the canonical cortical microcircuit, and yet we see remarkable variation in these gene expression patterns across cortex. And in particular, if we look at the dominant pattern of gene expression variation, what we ended up with was kind of a sensory to association gradient, a hierarchical gradient from sensory association cortex, which in this case was quite low dimensional and could be captured by a structural neuroimaging map, the T1, T2 map, and since we're highly correlated. And so the kind of takeaway from this was that we see specialization of cortical microcircuitry along a dominant axis here, which is related to cortical hierarchy and may contribute to the functional specialization of these hierarchical cortical regions. So that's one source of data that we're using. And then the other is really thinking about building dynamical circuit models of human neuroimaging data. And so the idea here is that we're going to be simulating, in this case, cortical microcircuits at some coarse level of approximation, where here we have interactions between local excitatory and inhibitory neurons within each brain area. We're going to embed that dynamic within a larger network and have these local circuits interact through long-range projections between cortical areas. And then what we're trying to model is the brain-wide patterns of functional connectivity in resting state fMRI primarily. And so that's going to give us some, for example, a target matrix about the patterns of functional connectivity, the correlated fluctuations in activity. We're going to, in our mechanistic model, embed these local circuits in a structural graph, in this case determined from diffusion tractography, which gives us kind of a structural scaffold on which the dynamics evolve and the regions interact. And that gives us a simulated model functional connectivity. And then we can fit the parameters of the model corresponding to neuronal and synaptic processes in order to maximize the fit to the empirical functional connectivity measure. And previously, we had targeted that from the perspective of this cortical hierarchy again. And here we were comparing a model in which each local cortical region was homogeneous and had the same parameters for excitatory and inhibitory coupling to one in which we had heterogeneous values across the cortical hierarchy in terms of the local recurrent strengths. And what we found when we fit that to, in this case, healthy data from the Human Connectome Project in terms of functional connectivity, and when we compared the model fits to the empirical, is that we could substantially improve the goodness of fit by including this axis of hierarchical heterogeneity in the local circuit properties through increasing local recurrence. And so this is simply to indicate that we can model heterogeneous local properties across brain regions and fit those to imaging data. Okay, so now I'm going to jump into how we can actually identify kind of a test bed to explore whether these mechanistic models can actually be applied in the context of pharmacology and disease. And so the idea is we're going to take this large-scale model, which has some coarse level of biophysical detail, and then, you know, at that level of the microcircuit properties, we can think about a disease process or pharmacology in terms of acting on different receptors. And so we have two pieces of information that we can put into the model. One is the brain-wide distribution of those receptor densities, which we can take as the proxy via the gene expression topographies from the Allen Human Brain Atlas. And then from the slice physiology literature and more microcircuit level experiments, identify which cells and receptor synaptic receptor sites are differentially engaged in specific receptors. So we have both kind of microcircuit level specificity as well as regional level specificity in terms of how receptors are differentially impacting the circuit dynamics. And then what we want to do is then make differential predictions at the level of human neuroimaging that we can test and constrain the model. And so, you know, in this endeavor, we're obviously trying to span many things. And so what we needed to start with was a very good kind of test bed where we had some decent idea of kind of what the ground truth ought to look like. And so for this, what we worked with our colleagues in Switzerland on a data set of resting state fMRI under LSD. And so in this case, what we're quantifying in terms of the neural measure is this measure we call global brain connectivity. It's just the average functional connectivity of a given brain region to all the other brain regions. And when we look at how that is altered under this serotonergic modulation by LSD versus placebo, we get this change in functional connectivity. And what you can see here is this you know, striking topography, and in particular, again, a kind of a sensory dissociation gradient with a very strong increase in visual cortex in particular. And the really unique thing about this data set in terms of a test bed to explore this modeling was that they were able to completely block both the neural and the behavioral effects of LSD by pre-administration of ketanserin. And so LSD is, you know, has kind of unknown pharmacology in terms of precisely which receptors are contributing, be it serotonergic or dopaminergic. There's kind of a few that are thought to be implicated. But in this case, we were able to really identify this neural pattern of the LSD to the 5-HT2A receptor because the catanserin is a more specific antagonist. And so you can see that here, where if we combine the LSD with catanserin and then compare that to LSD alone, we end up with a very similar topography, indicating that the LSD plus catanserin is very similar to the placebo alone. So in terms of now linking this to the gene expression, this gives us a very strong hypothesis, right? Which is that this pharmacological imaging map should actually align in terms of the regional topography with the expression of the corresponding gene HTR2A. And so to examine that, we again took this measure of changes in resting state fMRI from pharmacological imaging in Switzerland to these postmortem brain gene expression patterns from microarray measurements, and then looked at the correlation between these maps, just simply as that. And again, when we look at different kind of candidate dopaminergic and serotonergic genes and whose receptors are implicated, proposed for LSD, again, what we found is that our a priori target of the 5-HT2A receptor, the HTR2A gene, had the highest positive alignment with the neuroimaging map. And that was the one that obviously we had prior evidence for from the catanserin experiment. And so here you can look at the distribution. Here the gray plot is across all 10,000 genes in the Allen Human Brain Atlas, and see where all of these candidate genes kind of lie within that distribution in terms of the similarity of the gene to the imaging map. Okay, so this gives us, some confidence that the fMRI measures can actually be linked in terms of their topographic information to the underlying topography of the receptors, which are being targeted by the pharmacology. And so what we wanted to do in terms of the modeling is to now take this modeling framework where, again, we're simulating local EI interactions, coupling between brain regions, and simulation of the hemodynamic signal underlying the neuroimaging measure that we're measuring. And then the logic of how we're going to fit this to the pharmacology is as following. So from what we know about more detailed microcircuit physiology in terms of the effects of the 5-HT2A receptor, it's thought that these receptors modulate the gain, the transfer in terms of input current to output firing rate of excitatory inhibitory neurons. And so our free parameters that we're going to be fitting are the excitatory and inhibitory gain. And so we'll allow differential changes in gain for the two cell types and allow that to be a fittable free parameter. We're going to constrain the topography of that gain change to follow the HTR2A expression data. And so therefore the strength of the neural gain modulation is going to follow the topography of the gene expression. And then we'll get model simulated measures and compare that to the empirical fMRI. And that's what we'll fit to. Okay, so that's the logic. And then when we do the fitting, we can again look at what's plotted here on the heat map is the similarity of the empirical change in GBC, the change in mean functional connectivity, and the model change in GBC, change in mean functional connectivity. And what we find is that we get a maximum fit. Well, first of all, things are kind of collapsed. We see this kind of ridge-like structure. And what that means is that the kind of key effective parameter in the model is actually the change in EI ratio. So if you give a concomitant change in the gain for excitatory inhibitory neurons, it's the kind of net effect on excitability in the circuit, which predicts the change in the measures. And so what we find is that we get the best fit when this measure is positive, which means that there's a net increase in EI ratio. And that's, again, consistent with, I think, more neuronal level physiology, where we see changes in excitability in pyramidal neurons under this 5-HT2A modulation. So now we can look at the topography of the fitted model. So again, we get the preferential increase with an elevated EI ratio, and I think that's consistent with prior literature. And now we can compare these topographies, which we're fit to. So we have the empirical pattern of the delta GBC, and this is what we get for the model fit, and they're quite similar. We get a correlation of around 0.73. And the key thing here is that, what I showed you a few slides ago was the comparison of the empirical fMRI measure to the gene expression topography alone, and that was quite a bit lower. And so the kind of key takeaway here is that, yes, we can relate gene expression topography to neuroimaging itself, but we can actually improve our fit by simulating the types of neural activity and the impact of modulating that receptor on the actual neural measures that we're collecting empirically. And so by using the model as a bridge, we're able to improve the fit. Okay, so we can just kind of further characterize this in terms of functional networks, and we see that the model fits reasonably well. In particular, empirically, we get this preferential increase in connectivity for sensory networks over association networks, and that's something that the model kind of recapitulates. In particular, this increase in visual cortex of connectivity, as I'd shown, which is, again, driven by the higher expression levels of the HTR2A gene. Okay, so we can kind of do a few things to kind of validate and compare this. One is that we can compare the fit of our kind of a priori gene map to random shuffled maps. And you could just randomly shuffle the gain modulation randomly, but this is actually not really fair because this isn't really a plausible map because it's not smooth. You've broken all the autocorrelation. And one kind of technical thing that we had to solve was how to generate randomized spatially autocorrelation preserving surrogate maps that are still kind of randomized in their topography. And we have a method to do this if anyone's interested. And so then we can compare our HTR2A map to the gain modulation following a different topography, a randomized but still smooth and autocorrelated topography, and test for significance that way. We can also compare the HTR2A to other candidate receptors. As I mentioned, people have implicated in prior studies different serotonergic and dopaminergic genes and receptors. And so in this case, we can do this comparison for all of them. What you see is that the model actually has a kind of bifurcation-like dynamic where there's this kind of fringe along this middle ridge, and that ends up being somewhat of a spurious fit and therefore not very robust. And so one way that we can actually evaluate this is in terms of not just the peak fit, but the robustness in terms of the neighborhood in which it is fit. But in this case, what we see is that the HTR2A has the best fit of other candidate genes, and in particular has the best fit while also having a very high robustness, whereas the others achieve a fit through this kind of unstable, non-robust fitting near the bifurcation. Okay, so what I've shown thus far is basically fitting to the mean, but what we want to ultimately get to is fitting at the individual level and understanding individual differences in neural activity, different individual differences in response to pharmacology, individual differences in behavior and symptom responses. And so in this case, where we have 24 subjects, we can look at the mean map, but we can also look at the principal axis of variation. So what we do is principal component analysis, PCA, on this matrix of subjects by brain areas. And this is saying, what is the spatial topography by which this neuroimaging GBC measure changes most across subjects? And what's kind of interesting in that is that in this case, it's very similar to the group effect, which indicates that the dominant way in which individuals differ in response to LSD is essentially a change in intensity along the same mean pattern. And because of that, when we look at that in the model, and we've fit the model now at the individual subject level and do the same PCA analysis, the model obviously has somewhat constrained degrees of flexibility and degrees of freedom. And so the model, again, preferentially varies in a similar topography to the mean. And because empirically we observe a similar pattern, we can claim that the model is able to actually not just fit the mean change under the pharmacology, but also has sufficient expressivity to capture the dominant pattern of individual variation in the empirical measures. And so we can, again, kind of look and see in the empirical and model data, both of them are kind of dominated by a single axis of variation in terms of the first principal component. And then we can compare how well individual subjects, each dot here is the individual subject, is fit by the group level mean, the group level fit, or the individual level fit. And what we can find is basically that about half the subjects are very well fit with a consistent pattern, and are therefore well fit by the mean, but there are also a number of subjects which are not well fit by the group. And so that's a very interesting thing to study in the future, is what are the aspects of heterogeneity there as well. And so the last thing that I want to end with is how we can now hope to kind of bridge this all the way to behavior. And so the way that we do that in the data is by looking at individual symptom dimensions and then how the neural measures relate to individual differences in the symptoms. And so in this case for LSD, what we can look at here is 5 kind of categories of symptom ratings, essentially. And you can see from the error bars, there's large variation across individual subjects. And so what we can do is across these 24 subjects, create these types of maps, where this is the map for each brain region. We're saying, what is the correlation, or in a regression, what we call kind of a beta map, between the symptom measure, in other words, the change in disembodiment score per subject, related to the neural measure, the change in the GBC measure per subject. And so here, these warm colors would mean a stronger correlation between, stronger positive correlation between the neural activity and the symptom score, and the cool colors would be a negative correlation. And so now we get, obviously, kind of interesting maps, which actually are different for different symptom dimensions. And the interesting thing is that they differentially relate in terms of how that map is similar to the empirical PC1. So that is the empirical map of individual differentiation in the neural pattern across subjects. In this case, for example, the disembodiment measure map is highly correlated with the dominant pattern of neural variation across subjects, but the other measures are less so. And so this suggests that when we're looking at biomarkers related to pharmacology or disease, it's not simply enough to say there's a mean neural change and a mean behavioral change, and therefore those must be related. It could be that the individual variation within the neural change is only partially related to some of the behavioral variation in kind of a higher dimensional behavioral space. And so in this case, we see something very similar now in the model, that the model PC1 is how it's a similar topography is capturing individual differences in the disembodiment scale, but not the other scales. And so I think this just kind of nicely illustrates the utility of looking at individual variation in terms of multidimensional differences in both behavior and neural activity. Okay, so to kind of wrap up, I've hopefully shown how we're trying to make steps to kind of bridge all of these different levels, from molecules to regional differences in receptor distributions to modeling circuit dynamics, constraining that with pharmacological imaging, and ultimately trying to relate that to symptom variation. And so we have these kind of different measures, and the modeling, I think, serves a very nice role to kind of try to link these levels of analysis. And so again, we can return to this question of, if we have a molecule, how do we kind of map that to symptoms? And this perspective of looking at circuits and systems suggests that we can, look at the regional pattern of variation for the molecule, for the receptors targeted by the molecule, and then think about individual subjects or symptom dimensions, which in turn map onto that same brain-wide regional topography and profile. And conversely, if we don't have a therapeutic, by identifying the regional kind of circuit patterns of alteration in terms of neuroimaging, we can then, for example, scan across different molecules which happen to align or misalign with the topography of brain circuits which are altered in that disorder. And so there's kind of this way for these things kind of meet in the middle, where neuroimaging and kind of circuit level focus can play a role. Okay, that's all I have. Thank you for your attention.
John, that's really wonderful, and is really a statement of aspiration for our entire field. Really, I mean, to be able to go beyond charts with means to really have individually meaningful biophysiologic markers that will allow both for drug discovery, but also for individualized precision drug application. It's inspiring.
Thank you.
And to go now to your last level, which is sort of behavioral, albeit not at an individual level, but nonetheless at a group level. Anita Clayton will be talking about an orally active neurosteroid that's related to allopregnanolone, very strongly related to allopregnanolone and to the brexanolone, which was the intravenous form of allopregnanolone that was FDA approved for postpartum depression. So we'll be talking about now the application of this new molecule in treatment of major depressive disorders. Anita?
So I'm really excited to be presenting clinical data as relates to a lot of what we heard about today. This is the phase 3 multi-center double-blind randomized placebo-controlled study evaluating the efficacy of zuranolone, which was SAGE 217, in the treatment of adult patients with major depressive disorder. And these are my disclosures, which generally have to do with treatments for depression and also for sexual dysfunction and disorders. So we've already been talking today about some of the factors that are pretty important, but there's really obviously no single biological or environmental factor responsible for the onset of depression. So I think we really need to think about genetic and epigenetic susceptibility, and then the impact of additional environmental triggers. And those are going to alter some of the biological mechanisms, some of which we talked about: stress, altered neurotransmission, etc., leading to dysregulated brain network function and subsequently symptoms that are individualized. We've already talked about GABA as the major inhibitory neurotransmitter present in 30 to 50% of all neurons. The important thing that I'm going to be talking about is the GABA-A receptor, which we've also talked about, and that family is really pretty complex in physiology, being found in different brain regions and neuronal circuits. The pharmacology, because there are 19 different receptor subunits, and the function, because there are differing synaptic locations manifesting as phasic and tonic inhibitory tone. Also talked about. This to me has been very fascinating to think about. There are essentially two synaptic GABA-A receptors. There are synaptic GABA-A receptors, which is where we think of as GABA binding and the benzodiazepine binding site. But there's also an extrasynaptic GABA-A receptor, which is the positive allosteric modulator, or PAM, binding site. The former, the synaptic GABA-A receptors, have to do with phasic inhibition, and it's mediated by these fast activation and related to synaptically released GABA. The extrasynaptic GABA-A receptors modulate the tonic inhibition because it's depending on sustained activation of both the peri and the extrasynaptic GABA-A receptors. And this is related to ambient GABA in the extracellular space. And I think what we're really talking about is both phasic and tonic inhibition. So we're talking about allopregnanolone. You heard about that before, related to brexanolone. It seems pretty clear that abnormal GABAergic function is one factor that may contribute to depression. Now, allopregnanolone is an endogenous neuroactive steroid. It's a potent PAM of GABA-A receptors, and decreased allopregnanolone CSF is correlated with greater HAM-D17 susceptibility and severity. So when we think about decreased allopregnanolone, we could potentially assume that this has potentially, I've been sitting here a long time today. So decreased allopregnanolone in the CSF really can be correlated to greater severity of depressive symptoms. Now, zuranolone, as I mentioned, was SAGE-217. It's an investigational oral neuroactive steroid, GABA-A receptor, PAM, binding to both synaptic and extrasynaptic GABA-A receptors. And it's distinct from benzodiazepines. Those are the ones, as we talked about, which are really just phasic and binding to synaptic GABA-A receptors. The pharmacokinetic and pharmacodynamic profile really supports once-a-day dosing orally, and it's clear that it has potent activity at GABA-A receptors in vitro, and plasma concentrations are increased when zuranolone is taken with food, although it doesn't matter whether it's a high or low-fat meal. This is a very comprehensive program trying to look at GABA-A receptors, and in particular zuranolone, in both postpartum depression and in major depression. And we're not talking about treatment-resistant depression. We're talking about general run-of-the-mill depression. Some of these studies have already been completed. The Robin study, I'm going to mention briefly, in postpartum depression, major depression treatment in the phase 2 study using 30 milligrams versus placebo. And then I'll be reporting on the Mountain study here, which is the phase 3 using 30 and 20 milligrams versus placebo. So this is the postpartum depression study. This involved women ages 18 to 45 who were within 6 months of delivery. They had a postpartum depression, meaning they had a major depressive episode where the onset occurred between the beginning of the 3rd trimester and within 4 weeks of postpartum. And they had to have a HAM-D rating scale total score of greater than or equal to 26. These women were randomized in a 1-to-1 double-blind fashion to receive placebo or 30 milligrams of zuranolone. They received oral treatment in the evening with food for 14 days, once-a-day dosing. And the primary endpoint was the least square mean change from baseline in the HAM-D17 total score at day 15. And there was naturalistic follow-up for 45 days. And these data look much like what you saw with Jamie Maguire's presentation related to brexanolone, except the time frame is extended because this is an oral medication administered over 14 days rather than an IV administration over 60 hours. But you can see the first measured time point was at day 3, so after 2 doses of the medication. And there's already a drop in the HAM-D score of 12.5 points, and it differs significantly from the drop with placebo. Also at day 8, so after a week of treatment, significant differences and further drop. And then at day 15, the primary endpoint, a drop in the HAM-D score of 17.8 and statistically significantly better than placebo. That was also shown to be true at day 21 and out at the 45-day mark, where the HAM-D reduction was 19 points with the zuranolone. This is pretty amazing, and you can see so that there appears to be this very rapid response, perhaps the switch being flicked, and then it continues and it's really sustained. If we look at the adverse effects, the rates of any adverse effect with zuranolone was about 60%, and with placebo was 52%. So really not very different. The most common adverse effects seen were somnolence, headache, dizziness, diarrhea, and sedation, frankly. But most of these side effects were considered to be mild or moderate, requiring reduction in doses in a very limited number of people, and only 2 serious adverse effects that really did not indicate any signal. Now, the study with zuranolone in major depression was designed in a similar way. Patients were men and women, both in this case, with unipolar depression for greater than 4 weeks. And their HAM-D total score needed to be greater than or equal to 22. Again, randomized in a 1-to-1 ratio to placebo or zuranolone 30 milligrams. The first 7 days were administered inpatient in this particular study, and the second 7 days were then administered outpatient after seeing that this was tolerated reasonably. Now, people could not be part of this study if they had a history of suicide attempt or had treatment-resistant depression, or if they had initiation of psychotropics or a change in those within 30 days prior to screening. And again, the primary endpoint was at day 15, the LSM change from baseline in the HAM-D total score. And again, you see a really rapid response here. In this study, they measured at day 2, so really after a single dose. And so the drop was not as great, just a little less than 6 for zuranolone in the HAM-D score. But then at day 3, it's down to about 9. And you can see, again, this rapid decline, all significantly different from placebo. And to day 15, where the decrease was about 17.4 mean score on the Hamilton Depression Rating Scale. So this is the primary endpoint, which was significant. It was also significant a week after the last dose administration and 2 weeks after the last dose administration, but not sustained completely out at 42 days. Still, a fairly robust continued response. Again, no serious adverse effects. Patients receiving zuranolone, about 53% of them had any adverse effect and 45% in the placebo group, and very much the same adverse effects: headache, dizziness, nausea, somnolence, sedation. Very similar to what was seen in the patients with postpartum depression. And then this is the study we're going to talk about today, the phase 3 trial in major depression using zuranolone. Now, the study was really double-blinded until day 42, but in this study there was a naturalistic longer-term follow-up out 6 months, and I won't be reporting on those data today. Again, men and women were included, 18 to 65 years. They had to have a SCID diagnosis of major depression, and they had to have a score on the MADRS of at least 32 and a score on the HAM-D of at least 22. They were permitted to be on antidepressants that were stable at baseline, and about 30% of the patients were on antidepressants and remained on those through the study. Again, exclusion criteria included an attempted suicide in the current episode, uncontrolled medical conditions, currently taking benzodiazepines and other GABA-A modulators, or non-GABA anti-insomnia medications. They could also not be treatment resistant. They could not have failed 2 or more antidepressants in the current episode. So again, this is not treatment resistant depression. The primary endpoint was the same at day 15. And and these are the demographics. basically what you would expect in clinical trials of major depression, except there was a larger population of Black or African American individuals and a smaller compensatory number of white participants. Weight was pretty much around 21 kilograms. Their baseline HAM-D scores were 25.8 or 9. And as I mentioned, about 30% were using antidepressants at baseline and throughout the study. Unfortunately, it did not meet the primary endpoint for the HAM-D score change from baseline at day 15. And we'll talk about this a little bit. There were statistically significant differences from placebo with 30 milligrams of zuranolone also at day 3, where there was again a rapid drop after 2 doses, at day 8, so after a week of treatment, and at day 12, but not at day 15. The 20 milligram dose, which was a lower dose than was used in the previous 2 trials to potentially look for effectiveness at a lower dose and maybe better tolerability, really wasn't associated with better tolerability, and it was ineffective. It did not separate from placebo at any time point. So again, the 30 milligram per day was positive for a number of time points between day 0 and day 15, but not at day 15 itself. So to try to figure out what was happening, there was a post hoc analysis that was done that looked at measurable drug concentrations in the patients. And what was found was that about 9% of patients taking 20 milligrams per day and 9% of patients taking 30 milligrams per day did not have any measurable drug in their system. For those patients who had sorry, I skipped one. Let me go back to that, John. So a post hoc analysis was done in the Mountain study that showed about 9% of the subjects receiving the 20 milligrams per day and 9% of those receiving 30 milligrams per day of zuranolone had no measurable drug concentration in their system. And this basically said that they were non-compliant with the dosing of zuranolone. When these patients were excluded from the primary data set for the 30 milligram per day dose, it resulted in statistical significance at all time points. So at day 3, day 8, day 12, and day 15. When additionally, the patients who clearly took the drug and had an initial HAM-D17 score of 24 or greater, 30 milligrams of zuranolone was associated with a mean reduction of 14 in the HAM-D17 total score compared to 11.4 for placebo at day 15. So this is a drop from what you can see here, and when just considering the individuals who had measurable drug in their system. Slightly greater severity of HAM-D also contributed to an improved response. Again, pretty low rates, in my opinion, of an antidepressant in having side effects or adverse effects. So 54% in the zuranolone 30 milligrams per day group, 50% in the zuranolone 20 milligram group, and 49% in the placebo group. Again, headache, dizziness, somnolence, fatigue, diarrhea, sedation were the most common adverse effects. So no new signal was seen, and no signal for suicidal ideation or suicidal behavior, and nothing in laboratory values or vital signs. So zuranolone met its primary endpoint for the phase 2 major depressive disorder study and the ROBIN study in postpartum depression, achieving statistically significant change from baseline in the HAM-D total score versus placebo. And this was the 30 milligram per day dose. The Mountain study did not meet its primary endpoint of change in the HAM-D17 total score from baseline to day 15. There were statistically significant reductions with 30 milligrams per day of zuranolone at earlier time points, day 3, 8, and 12. And the 20 milligram dose did not separate from placebo. So it appears the 30 milligram dose is the minimal effective dose. And then in a post hoc analysis that excluded patients with no measurable drug concentration, statistical improvements were observed at all time points, including through day 15. And greater severity of depressive symptomatology further enhanced that level of improvement. So zuranolone was generally well tolerated, had a very similar safety profile as you might expect with things like headache, dizziness, and somnolence or sedation. But these were mild or moderate and well tolerated with minimal need to reduce the dose or with dropouts. So these data support the investigation of higher doses of zuranolone, doses higher than 30 milligrams per day in future clinical trials, and continued development of neuroactive steroid GABA-A receptor PAMs in postpartum depression and major depressive disorder. Thank you.
Great, Anita. Thank you very, very much. I'd like to take the prerogative of opening the questioning, and I'd like to pose first a sort of just a thematic consideration, and then ask a specific question to each of our panelists. So the question that I have is one that was really implicit in what Jamie was mentioning, and that is, if we think about excuse me, about behavioral disorders, not in terms of the constitution of the particular state, but rather in the kinetics by which the state changes occur, right? So just more broadly, one can think about PTSD as a disorder in which one suddenly has a particular kind of behavioral state that's characterized by autonomic hyperactivity, arousal, by fear, by diaphoresis, so and by specific kinds of affective accompaniments, or panic anxiety syndrome, or OCD, sudden intrusive state characterized by anxious symptoms, or in the case of depression, then an inability to be able to terminate a particular state, right? So that you're stuck in an affective state. And the role of the treatments, like Anita is describing, would be ideally that of being able to enable an individual to switch out of that state, because it's our normal human condition to be able to experience different states. Right, so that's, you know, the morning run or the evening cocktail or sex, drugs, and rock and roll. You know, there are all ways of being able to change our behavioral state. So what I would ask to each of you in a way is how you explain some element of the kinetics of the process that you're talking about. So my question first to John would be, how do you understand and model some of the scalar or fractal properties of the network interactions that you're describing, right? Because if you're looking at fMRI, you're looking at an aggregate measure over a long period of time, but presumably there is much more rapid kinds of interactions that are taking place and that constitute dynamic functional connectivity, as opposed to a 10-minute scan in which everything is averaged. And under those circumstances, then, can some of the behavioral variability that you talked about across individuals actually reside some of either the topography of the different global states or in the nature of the interactions between those states at a much shorter, more rapid time interval than that in fMRI. So I'm gonna hold that there, and then I'm gonna come ask each of you the questions, and then you can all ask questions of each other. So the thematic question to Jamie would be, why are there persistent effects? I mean, if you're able in a chronic stress model, whatever the changes are that occur, why are you, if you acutely are able to prevent the behavioral effects or to reverse the behavioral effects, why does that improvement and the network interactions that are associated with that sustain after the drug is no longer present? And for Etienne, a variant of that, which is if your alpha-5 modulating compound is tuning the circuitry in such a way that you are getting better signal to noise ratio, why is it that you're getting the neuroplastic effects, the changes in neural proliferation that you observe, unless you're inferring that there is otherwise at baseline a consequence of if you will, excitatory neurotoxicity that is again resident, let's say, in an older animal, but it's hard for me to understand unless there was unless you were actually, as you were describing, as a result of increasing inhibitory tone, altering neurogenesis, which is what you were describing. And it's the connection between the tuning of the circuit and the sustained change in neural architecture is something that would, for me, be helpful for you to re-explain. And then finally, for Anita, a question which is a very difficult question, but as somebody who is involved in clinical trials, you in the last slide that you showed on the effects of 217, you showed that there's a 12-point drop with placebo, right? Which is a very large drop. So this bedevils most of the trials for antidepressant drugs of any kind. But how have you come in the course of your work to understand what it is that allows a placebo to produce such long and, in that case again, enduring effects on something that presumably was already resident for a considerable period of time before the people came into the trial? So those are my questions. So John, since you've had the longest to think about it.
Yeah, so if I'm going to maybe interpret your question in terms of one of timescales and dynamics, spatiotemporal dynamics that evolve across multiple spatial and temporal scales. And I think that that's a very good point, and there's not a clean separation. In terms of the computational modeling, there's not going to be kind of one level of model which necessarily speaks to or explains all of these scales, and the degree of kind of resolution and complexity that we put into them in large part comes down to what degree we can kind of constrain it and inform it and fit it with different types of empirical data, right? And so the very simplified models that I was showing of just basically kind of neural mass, mean field activity is kind of well suited to this bulk measure of resting state fMRI connectivity, right? In terms of how we can go beyond that through different modalities. So this is actually one thing that we're very interested in and have published on a little bit in using, say, fMRI to look at the spatial topographies, but then using something like MEG or EEG to constrain something about the faster spectral temporal dynamics. And so we actually did that when we were looking at the sensory dissociation kind of hierarchical gradient and observed in resting state imaging a similar sensory dissociation gradient in spectral power properties. And so my real hope is that this also gives us a way to think about putting more complexity into the models. So for example, we had talks about different inhibitory interneuron cell types, which have different GABA receptor subunits, which have different timescales. And so that may be manifest in the circuit in terms of parvalbumin interneurons involved in gamma oscillations compared to SST involved in theta oscillations. And so maybe we can, kind of constrain and kind of test more detailed microcircuit all the way at the level of human imaging by bringing multiple modalities to bear on these questions. That's one way we're thinking about it.
Fabulous. Thank you, John. Jamie?
Yeah, so I just wanted to touch on a point that you made that I think is really important, is that when we're talking about these transitions between states, we're really talking about just normal physiological changes that everyone experiences, and trying to understand how people do get stuck in one state versus another. And we're focused on depression, and so we're focused on that negative pathological state. But these are all not normal transitions that we're trying to understand. And I think we may have been conceptualizing it different, but we've been doing this for a long time. We've been using chronic stress to model depression for a long time, and using that really to push the animals into a different behavioral state by using chronic stress. And so now we're really looking at the network level to understand how those transitions are mediated. And importantly, when you do push an animal or a person into that negative pathological state, how can you restore the healthy state? And when we're thinking about the persistence of these changes, because that's really something that's been at the heart of this and really fascinating us, I think that question is a lot easier to answer in animal models. When we push them into the unhealthy state, or we push them into or we restore the healthy state, I think experimentally we can maintain those animals in that state for a more controlled amount of time because their experiences are so limited. So I think clinically, when we're looking at these prolonged antidepressant effects, I think at some point, most of these patients are going to be pushed back into the unhealthy state. Another episode is going to arise. And that the temporal dynamics of that are going to be different for every patient, depending on their underlying neurobiology, their experiences, their environment. All of those things are going to come to play. So I think that we can try to understand some of these things at the preclinical level in a much more controlled environment. But I think some of those concepts are applicable to the clinical setting. It's just going to be a lot variable, and then things like environment and experiences really come into play.
Great, thank you, Jamie. Etienne?
So why do we have this neuroplasticity effect with increasing GABA function? So I'll answer that hypothetically because we don't have the exact answer yet, but we actually just put a couple of grants to actually look exactly at this why, the mechanism. So there's a conceptual level, and then there's a more mechanistic level. What are the molecules that mediate? At the conceptual level, there are two aspects. One of them, yes, we know whether it's in context of chronic stress or in the context of aging in this case, we know that the system is downregulated. We know that the presynaptic, which are the somatostatin neuron, are affected, the GABA function is down, the structure is down, and we know that postsynaptically, the pyramidal cell dendrite have shrunk. So the pathway is down, and it's reflecting reduced signaling, reduced filtering of incoming information. So it would make sense that we're not just boosting GABA overall, we're actually just restoring GABA in the dendrite, allowing for better discrimination of signal. So in a simple way, you could say, The system works better, it recognizes it, therefore it strengthens it. Basic neuroplasticity event. You use it better, you grow it. Same as the muscles. This would be a conceptual aspect. It makes sense rather than just, a benzo would not do that because it would dampen, it would break down that synchrony of events that is needed for proper signaling. And it's interesting, we see that postsynaptically. Again, the two elements, what we know now is that postsynaptically, we have a rescue of the phenotype. Those neurons grow back, those synapses, we have more spine. We don't know yet whether we actually rescue the presynaptic phenotype. We bypass. With the drug, we go directly to the receptor. We don't actually mess around with the somatostatin neuron. There may be some feed forward, increase activity, increase forward inhibition. We don't know yet. We haven't looked. We have some hints that it may not, at least within the time frame we looked, restore the structure of the somatostatin neuron, and you need sustained activity of the drug to continue, but we have to tell. At the mechanistic level, there's some interesting aspects of the alpha-5 GABA-A receptor. The kinetics are different from others. You know that if you give a benzo, activates the alpha-2, for example, quite strongly, and then you get desensitized, you get internalized, the whole pathway. The alpha-5 doesn't do that. Actually, it's shown that if you give anesthetics, which, the really extreme, you have sustained increase of alpha-5 at the membrane. So somehow the kinetics are different, so you don't desensitize the system. And actually talking about tonic versus phasic, the alpha-5 does both. It's classically known as mediating phasic or tonic inhibition, but it's actually been shown recently that it mediates maybe up to 70% of phasic inhibition in dendrites. T. Jacob at Pittsburgh has shown that if you activate this receptor, you actually shift from the synaptic to the perisynaptic, so from phasic to tonic inhibition. So I'm giving you all this information. The point is we don't know yet, but there's some really nice mechanistic cues that we're getting from the specificity of the alpha-5 versus others that need to be investigated. And it's exciting because it's really kind of uncharted territory. It's an unexpected finding, and these are sometimes the coolest, they give you new ideas.
Terrific. Thank you, Etienne. And Anita?
Well, I don't know if any of us can answer questions about placebo responses, but I think that there are some things about a drug in which there's an anticipation of an acute response or potentially actually having an acute response that may do something different in terms of the biologic response that is a placebo response for people. And, if you think about phasic effects at the GABA receptor, when people get benzodiazepines, they feel better very rapidly. And that has an impact on, again, future anticipation. And we can imply that too in so many other ways because we explain the study to them. And they may actually from the drug, there presumably is this phasic response, but really what we're most interested in is the tonic and sustained response. So I think also we often don't think about a placebo response as a biologic response because we can't figure out who is what and why beforehand. But I know we did this study years ago and published it of taking women who had menstrually related migraines. So we knew when they were going to get a migraine, and we brought them in and we gave them sumatriptan or placebo, and we measured blood measures and metabolites of serotonin, very rapid, repetitive measures. And the sumatriptan, basically was this flat line because we'd administered a serotonin 1B, 1D drug to those people. But interestingly, the placebo responders biologically did what we did with that drug, which is they had significant increases in serotonin but then and that was sustained over about an hour. But by the end of an hour, when their migraine was gone, they were back to the levels that of the people who got sumatriptan and et cetera. And so if we hadn't done rapid measures, we wouldn't have seen that. More interesting is the placebo non-responders who actually had decreases in their levels of serotonin over the acutely and then over the course of that hour. But again, at the end of the hour, they were back where everybody else was. So maybe our bodies do things we don't even know we're doing, and we're not thinking, oh, I'm going to do that. But they have these effects. And then if you've impacted on the other factors, so not so much a physical response, but the environmental factors like Jamie just suggested, then that may make a difference. Because I think, you know, and we may have to repeat these treatments, right, at some extended period. So if we flick a switch and it's on, eventually the light burns out potentially, especially if you've got chronic ongoing stressors, which, you know, wear people down and reactivate that problem. And so I think when we do something very rapidly and quickly, or that's anticipated, we might see something very different in a placebo response. And remember, the placebo responders, some of those people in the placebo response are placebo responders. Other people are not. So the people who get a placebo response, somehow they're doing something that leads to what appears to be a similar kind of outcome.
So yeah, I mean, you raised 2 points there at least. I mean,
yeah.
One is the one again are the kinetics of the response in a placebo responders. I mean, we were classically taught that you have a placebo response and then it attenuates. But in the studies that you described, you know, the attenuation isn't obvious. And the other implication for me is that when John is really sort of modeling the network interactions and really from again, from the synaptic level, from the local field potential level up to the behavioral level, being able to do so in placebo responders would be every bit as interesting as doing it in people who respond to whatever, you know, whatever the active compound is. Because that's really what we're trying to do. If we could figure out, as you're suggesting, I think, Anita, if you could figure out what the biological mechanisms are that are recruited in some placebo responders that allow for sustained response, that would go a long way towards our really understanding the biology of the illness, and more importantly, how to get people the hell out of it. So, all right, I've asked my questions. So I don't know if you have questions that you want to ask to each other, or whether they're going to pull the plug on us. But please, by all means.
So I, just along the line of the placebo, I remember we're talking about the brexanolone study, where you're talking about an IV infusion, which has a much higher, you know, placebo effect, and also you're getting round-the-clock care and treatment, and so that alone has an impact on I think, you know, the buffering of stress and the transition to a healthier state as well. And so, yeah, I think, like you said, understanding that phenomenon is as important.
Yeah, I'd agree with you, by the way. I mean, put somebody in a hospital, stick an IV and give them something, you're going to have a high placebo response. There's no question. It made it, you know, just personally, to me, it made it all the more remarkable that there was a separation of active compound from placebo under those circumstances.
You know, a quarter of people who get narcotics have a placebo response. I mean, who think they're getting narcotics have a placebo response, right? So it's not only psychiatric drugs per se, or antidepressants.
Certainly not. I mean, Huda Akil showed that in her Science article. I don't know what that was, 40 years ago or so. You know, that you could block the placebo response with naloxone. So there's clearly an underlying allergy, but right now it certainly does seem to, you know, I mean, it obscures the effort to identify new compounds because, you know, it's the old cartoon of somebody standing with their foot in scalding hot water and the other foot in cold water, and they say they're standing in warm water. You know, I mean, we have these group effects, and it really prevents us from really being able to better understand the biology of treatment response. But you know, I mean, I think with some of the clues and some of the methods that you guys have described today, it may be less important to identify the mechanism going in, if you can, again, as John suggested, if you can identify the mechanism coming out, if you can actually see what it is that characterizes response, irrespective of what somebody got initially, that's going to enable you to develop more precise models of how to intervene.
So let me follow up with a couple of questions. One for John there. So, you know, if I summarize very crudely, you know, what I see the approach. So, you know, based on the one drug with a very specific known mechanism of action, you can use your approach, you know, through gene expression, pattern of expression to associate with some potential symptom dimension. And you showed a really fantastic example with LSD. Now, that's going to be only possible with drugs that are available in human, that can be tested in human in fMRI, I assume. Have you thought of starting rodent studies for that as well, you know, so you can look at experimental drugs? Because talking about, you know, what do you call a companion biomarker assay, that would be fantastic, you know. So what's your plan there?
Yeah, it's a really interesting idea. So we actually have a project that we're starting on that in the mouse, where we're using the Allen Institute receptor mapping in the mouse. And right now we're kind of starting that out with a study looking at fMRI of mice under neuromodulatory perturbation, and trying to see to what degree we can kind of model, again, through these kind of gain changes. In this case, actually looking at changes in the dynamics of the activity under neuromodulatory control. So we're trying to kind of move in that direction.
Yeah. And that, I mean, you could combine that with there's some drugs, you know, that target receptor that have very specific expression. I mean, in this, we take at alpha-5, you know, it's high hippocampus, frontal cortex. But, you know, you have some target that could maybe be used early to test that. But yeah, looking forward to, yeah.
I think in a rodent model is also a place to think about having kind of more detailed microcircuit models, say with multiple interneuron cell types, I think.
Well,
a big direction I'm interested in.
John, if you want some fMRI data from mice with allopregnanolone, let me know. I'd love to, to give you those data.
Yeah. I had a question for you, Anita.
Oh, okay.
Sorry, we're talking about a little bit of delay there. So the people who had, 9% of subjects who had no plasma level, I assume you ruled out any metabolism effect, any pharmacogenetic effect. Have you looked at that?
No, generally these are part of the lab studies drawn at various points, and we're really just looking for no measurable levels. It seems like, given the pharmacodynamics and pharmacokinetics, we really, you know, it's only requiring once a day dosing. We really should see some drug in their system. So no, so they haven't.
Okay, they just didn't take it. Huh? Yeah.
Yeah, they didn't.
Horrible.
And that seems like actually a low rate in a depression trial, frankly. But they were coming in very frequently. So again, another thing that might enhance the response, because we're seeing them frequently. I had one question for Jamie, and I'm going to have to get off in just a moment. Given that this is like affective states, I'm wondering if you think that similar effects could impact on the opposite state, so mania.
Yeah, absolutely. Especially, you know, we started, I didn't talk about this at all today, but we started getting into this from work on postpartum depression. And so we were very interested in the relationship between bipolar disorder and postpartum depression. And certainly, I would love to look at that. And it's just a matter of what we can reliably model in our animal models and how we would look at that. But I do think that that is something that we're looking only at one aspect of the state change. But the nice thing is that we're looking at it from two different ways. We're looking at perturbing it with stress, and we're looking at how we can restore it with now a clinically demonstrated effective treatment to come at it from the other side. But I do think that there's other ways to look at how that state becomes dysfunctional. And we've been really focused on GABA receptors and allopregnanolone, but as I alluded to, I think that there's a lot of different ways to perturb the network state, and getting at the heterogeneity in the patient population, that there's a lot of ways to disrupt that state. And so trying to understand then how we can restore that state. But I think that it's not, we're coming at it from the GABA receptor and chronic stress, but I think that we're trying to look more broadly at things that can disrupt network states.
Yeah, and I think that what this does, just to pile on a bit, is that it brings us back to what, Biff Bunny and Fred Goodwin talked about in the 60s when they were focusing on bipolar illness and said really the answer to bipolar illness will be found in the switch. In other words, once the state is instantiated, there are going to be all kinds of homeostatic adjustments that may really not tell you about the pathophysiology of the disorder. But when it switches from one state to another, if you could actually isolate that and study that process, that's where the biggest bang for the buck is going to be. And it's what led me to study, premenstrual mood disorders years ago, because you have a new switch each month, and so you can identify that period. And I will say also the studies that that everybody knows of patients with bipolar disorder who are subjected to a sleep deprivation, and work that we did in the intramural program 35 years ago. We took somebody who had a brilliant response to sleep deprivation. I mean, an intractably depressed patient who was all better. And we put in an IV and hooked up an EEG and put her back to sleep. And she awoke spontaneously 2 minutes after going to sleep and was completely depressed. And this is a patient who ultimately suicided. So so sleep itself is a huge physiologic trigger. And in women with postpartum depression, the one thing that we say to all of them is make sure that your sleep is protected. You've got to have help for that, because otherwise, particularly people who have had prior histories of postpartum depression, or if they're bipolar, they're at huge risk if their sleep is disturbed. So we do have these built-in physiologic means of regulating our state transitions that can go awry. John, I want to just ask a silly question, perhaps, but do you know, by the way, how good a measure of inhibition excitation, the slope of the power spectral distribution is in EEG? I mean, is that a reasonable, rough and ready measure of the excitatory and inhibitory balance?
Yeah, there has been some recent work on that, especially from Brad Wojtek's group at UCSD, which has looked at some of that in terms of modeling as well as analysis of different types of data. And I'm forgetting what manipulation they had, but it seems like there's kind of convergent evidence from multiple directions that show that the shift is reflective of that. I think that we still don't quite fully understand kind of the mechanism by which that is, but in terms of approximate, my impression from the literature is that it actually is quite interesting. And so that's actually one way to actually think about constraining data models with multiple modalities is through that.
And for Etienne, again, you didn't talk much about alpha-5 in depression. You spoke mostly about cognition. Are you pursuing that avenue as well?
Yeah, yeah, we have actually really nice data, rapid effects. You know, we're writing a paper now. It's different from what, you know, you hear about the rapid antidepressant effect of ketamine, which is really actually 24 hours after single injection. You know, here you have in models, you know, in the relevant behavior dimension, you can look at mouse, which has come kind of more like anxiety related, sometimes anhedonia-like, but we have very rapid effect as well. We have immediate antidepressant effect. So no, it's exciting. We have anxiolytic effects. It's interesting. We're writing a paper now on the chronic effects. You know, I showed you the effect on cognition in the UCMS model, you know, and you have a rapid sustained effect. And now we did the chronic exposure of the compound, you know, chronic treatment. And the effect is actually more robust on cognition than on the depression-related outcome. Mm-hmm. So that was actually interesting. So we're going to follow. Yeah, we're following up on that as well. It's quite interesting. Yeah. I think what's new is the cognitive aspect. Because you know, none of the antidepressants have any effect on cognition. If any, they make people dumb at times, and benzo definitely do that. Unfortunately.
Guys, I have to go. I have another appointment that I scheduled thinking we'd be finished by now. So sorry, I'm just going to leave and check out.
Thank you, Anita. I suspect that everybody else is going to have to go too, unless there are any, and feel free, Anita, actually. I don't know if anybody else had any remaining comments or questions.
This is a lot of fun,
yeah,
guys.
Thanks,
yeah, it
thanks,
was.
Anita.
Yeah.
It was great.